like to introduce Britton and Andrew Restrepo. They are and were students at Evergreen State College in Washington State. And their research is focused on vagrant lichens in the monument. And this research was advised by Dr. Lolita Calabria.
Give me 1 second to get this all kosher.
Andrew, if you could maybe move your video so we can see your face, but other than that, we can hear you just fine.
All right, can you guys see just the presentation mode? Everything looks good?
Yes.
All right. Hello, my name is Andrew Restrepo, and I'm here with my friend and project partner, Britt Glenn, and this is our report on the relationship between vagrant lichens and grass communities of the Cascade-Siskiyou National Monument. Our work presented here is part of a larger endeavor headed by John Bilella and Nils Nelson, which extends outward past the borders of the monument. So first and foremost, to those who are kind of new to this realm, which some of you might be, what is a lichen? Well, briefly, it's the result of a symbiotic relationship between a mycobiont, which is the fungal component, and a photobiont, which is more often a green algae, but sometimes a blue-green algae called the cyanobacteria, and even sometimes both. This relationship is based on the algae photosynthesizing and feeding the fungus while the fungus provides shelter for the algae. This shelter or body of the lichen is called a thallus. So even more deeper, what is a vagrant lichen? It's a lichen with a rare morphological adaptation that improves its distribution capabilities. This adaptation is the ability to subsist and thrive past the constraints of the substrate attachment. The species that produce vagrant growth forms are all of the Saxicolous or rock-dwelling variety. This morphological difference allows these thalli to become mobile across the landscape through wind and also rain events, but also through animal vectors. Due to this mobility, vagrants are prone to reproduction by fragmentation. They are also known to reproduce sexually through spores, but this is believed to be a bit more uncommon due to spore-bearing structures being relatively rare depending on the species. So first and foremost, why do this project? Well, vagrants appear to be pretty common or more common in other arid parts of the American West, such as the Great Basin and the shrub steppes east of the Cascade Crest. But the monument on the west side of the crest boasts relatively healthy populations of these vagrant growth forms. Though most are not considered rare on a species level, the unique morphology of the vagrants and their seemingly disregard for the traditional substrate is what sets them apart. These populations are considerably fragile and vulnerable, but at this time, the species are unlisted. Disturbances to these populations come in the form of human activity, trampling and grazing by cattle and wild horses, and the aggressive encroachment of non-native grasses into the region. This leads us to believe that conservation efforts of disturbed vagrant habitats should be strongly considered. And so we'd like to give you a quick rundown of the species we encountered. Before we do that, I would like to say that most of these pictures you're about to see were taken on an auto-montage Z-stacker microscope based at the Evergreen State College. So I'd like to say thank you to them for letting me weasel my way into that room. So this is Dermatocarpon bachmanii. This is probably the most common of the vagrants that occur in Southern Oregon and in the monument. It's a foliose lichen, meaning that it has both a distinct and lower and upper side. The rock-dwelling growth form of this species is partial to basaltic rock and grows most commonly in the seepage zones or where water runs along the surface of the rock. The more charismatic vagrant forms, like the one shown here in this picture, seem to thrive in both open, flat, arid, rocky areas such as the Pilot Rock region and areas with seasonal pooling such as Condi Creek and habitats off Highway 66. So bachmanii has two apparent modes of reproduction: thallus fragmentation, where lobes of larger thalli break off and form their own autonomous specimens. And this is thought to be the more common way for the species to reproduce. But they do have spore-bearing structures called perithecia, where the spores are produced and propelled into the atmosphere. The spores then attempt to colonize with a compatible algal partner to form a new thalli. It has yet to be fully determined which mode actually is the predominant reproductive strategy. And so here on the left, you can see the more flask-shaped perithecium structure. And on the right, which is a little more difficult to see, is the elongated asci, which bear 8 ascospores each. These ascospores are considered sexual spores due to the fact that they are offspring of 2 separate parent fungal hyphae. And this is Bachmanii in the wild in its 2 different growth forms. On the left is the vagrant growth form. You can see how densely these populations can occur. In some areas of dense population, the thalli are stacked 3 to 4 high on top of each other. This usually occurs in habitats with more depressions and vernal poolings, rather than in habitats that are more arid with flatter topography. And this is probably most likely due to the fact that drier, flatter habitats are more conducive to easier mobility. Now on the right is the same species when attached to what is called the mother rock. Though these 2 morphologically differing populations occur in direct proximity to each other, it's a bit unclear at this point how the vagrant and attached populations are related. Are they results of the sporification and fragmentation, a combination of both? It's kind of a chicken or the egg paradox question. One way to possibly answer part of these questions, though, would be to perform a population genetics analysis on vagrant and attached populations to determine whether they are actually genetically identical or not. And so this is Aspergillia californica var gigantea. This one is a rather funky looking fruticose vagrant. Fruticose meaning it has no distinguished upper or lower sides. Instead, the lobes of the thallus are filiform or thread-like, as you can see in this picture. As of now, there are only 6 known locations of the species variation, and they're all localized in Southern Oregon. And Gigantea has 3 known modes of reproduction. Firstly, the thalli are notably fragile and can easily fragment. And like other vagrants, these fragments are capable of becoming autonomous specimens. Gigantea also has two different spore-bearing structures, pycnidia and apothecia. Pycnidia are flask-shaped or spherical structures, like you see in the middle here, that produce and release pycnidiospores, which are asexual spores. And asexual spores are just the clones of a single parent hyphae. The rounded structure you see on the right is the apothecia, which produce and release ascospores, which are the same type of sexual spore that is produced in the perithecium, like we see in the dermatocarpon. And in the wild, these species can be a bit trickier to see, as you can tell on the left-hand picture. If not looking close enough, you could easily gloss over them thinking they were remnant plant roots or other random plant debris, especially when the thalli are in the earlier stages of their growth. On the left is an example of the incipient morphology, or the earlier growth stages. And on the right is the intricate vagrant morphology of a well-established and battle-hardened thallus. It's because of growth forms like that vagrants are sometimes referred to as tumbleweeds. And this is the yet to be identified Xanthoparmelia species. It's another foliose vagrant. And you can see that this foliose species has the incurled lobes like dermatocarpon, but the lobes form a more tubular shape. This species appears to be the more rare vagrant in Southern Oregon and in the monument, with only a few known populations having been observed. And due to the lack of any observable reproductive structures, this species at this time is thought to only reproduce by fragmentation, which brings up the question of how genetically diverse populations of this species are if they are only reproducing by fragmentation. If this is their only reproductive strategy, this would lead one to believe that population would be more genetically identical than not, and thus more at risk from any disturbance of the niche they fill in their habitat. And this is just a couple of pictures of them also in the wild.
So here are a few general characteristics of vagrant dermatocarpon and Aspergillia habitat. They're typically found in relatively flat, scabby areas, which are rocky with these red volcanic soils that are oftentimes basaltic. And they can be prevalent in shallow depressions. Like Andrew said, these can act as vernal pools, areas of seasonal water accumulation. And they also seem to prefer bare ground with relatively sparse vegetation. And this can be provided by these native bunch grasses like the fescues, which seem to leave space between the bunches for vagrant community establishment and movement. And these native grasses also seem to be important indicators of intact soil habitat that vagrants usually exist in, along with soil crusts and mosses. And so this background photo is of a site just west of Pilot Rock, which is a very large opening that has vagrant Dimetacarpon bachmanii throughout. And this photo was taken during our first round of fieldwork last fall. And this is a site south of Lincoln off of Highway 66 in a large flat area near the airport, where we also found vagrant Dimetacarpon bachmanii. And this photo was taken in the spring just a couple of weeks ago. And as you can see, the site is fairly flat, pretty scabby, with native bunch grasses and a good bit of bare ground. And we did notice some grazing disturbance in this area, but there was plenty of intact habitat with soil crusts and mosses. Here's another Dimetacarpon site near Condi Creek at the northern edge of the monument. And this habitat's similar, fairly flat and scabby. Where the vagrants are, there were native bunch grasses and bare ground, but here you can also note the moisture, especially towards the bottom of the photo. There was actually standing water here at the time, and it was kind of muddy. And so this site had some of the largest lichens that we found, and it was an area where they were sort of stacked on top of each other. So this seasonal water accumulation might help stimulate growth. This site is also south of Lincoln near the airport. And this is the third known Aspergillia californica var. Gigantea site in the world, basically. And it has, there's also a large population of vagrant Dimetacarpon bachmanii in this area. And it has similar habitat characteristics as the other site with a few more shrubs. It was also reasonably intact, but there was some grazing disturbance like the other Lincoln sites, which seemed to fragment the Aspergillia. And so this is the same area, just zoomed in a bit. You can see those native bunch grasses, the large amount of bare ground. And you can tell how well the Aspergillia really blends in with the rootlets and bits of dead grass and how easy it could be to miss. So this next one is zoomed in a bit more so you can see the Aspergillia a bit better and just notice how it's occupying that bare ground between the bunch grasses. And then this is just a few feet away, maybe 10, well, maybe 20 feet away. And there was a large population of vagrant Dimetacarpon bachmanii there also existing in a similar fashion on the bare ground between these bunches, along with some intact mosses and soil crusts. And so, whereas when we look at invasive grass cover, it's going to become a lot more matted and dense and can form that thick thatch, leaving very little bare ground. And so this seems to inhibit vagrant community establishment and movement. And also important, these vagrant grasses also seem to be indicators of disturbance. And so even in some areas where we would find a fair amount of bare ground with relatively sparse vegetation, these more disturbed sites usually lacked that intact soil habitat where the vagrants seem to exist. And so we found this to be an especially big problem down in the California side of the monument, where it seemed like wild horses had really trampled a lot of the habitat. And a few of the species that we commonly encountered of these invasive grasses were cheatgrass, medusa head, bristly dogtail, and poa bulbosa. So having observed some of these habitat trends and possible relationships, especially with the grasses, our research partners, John and Nils, wanted to conduct a more formal plot-based study with the objectives to examine and document possible relationships between native and non-native grass cover and vagrant lichen populations, and to catalog the habitat types that sustain vagrant lichen communities, with the hypothesis that non-native grass cover negatively impacts vagrant lichen abundance.
And so we followed some systematic and easily replicable field methods devised by John and Mills. Before heading out into the field, we would choose search locations via landscape level analysis through topographical map programs such as CalTopo, ArcGIS, Google Maps, based on known habitat types supported to vagrant populations, documented historical observations, and from previous successful field trips. Upon arrival to a prospective location, we would intuitively comb the area. We would discover a substantial population. We would then document a detailed description of the habitat, including slope, aspect, evidence of disturbance, vegetation, or lack thereof in the proximity. Next, we would determine where our research plot would be situated. We would establish the plot center in the area where vagrant ground cover concentration was the most dense. And from this plot center, we would measure out 30-foot transects in each of the cardinal directions. Going one transect at a time, we would use a 1.5 foot by 1.5 foot gridded microplot to document ground cover percentage along the transect every 3 feet. 10 microplots were documented per transect, and ground cover measurements were organized into 6 subgroups: vagrant lichens, moss, grass, forbs, rock, and bared ground. Estimations of ground cover for each microplot would total 100%. So for example, a given microplot would have 20% lichen coverage, 0% moss, 20% grass, 10% forbs, 25% rock, and 25% ground, all equaling 100. Native or non-native grass dominance was then determined by whether which grass type accounted for more than 50% coverage.
So here's a map that shows all the sites where we located and surveyed vagrant populations. We located 11 Dimetacarpon bachmanii populations and 3 new Aspergillia californica var gigania populations in and around the monument. All but one of these sites were newly discovered. And the Aspergillia sites actually represent only the 3rd, 4th, and 5th known locations of the species, which expands its range now into the monument. And I did find one other good Aspergillia site in the area, but it turned out to be on private property, so I didn't include the pin on this map. But one of the northern sites is actually on the Vesper Meadow Restoration Preserve, which is just outside the monument. We surveyed plots that Andrew was just talking about on 5 of these sites, including 4 Dimetacarpon plots and 1 Aspergillia plot. And this contributed to the 12 total plots of the overall study. So all of our surveying basically took place over 3 separate trips. The first one was last fall, and then we had 2 this spring. And so during our initial searches, we were mainly using aerial photography, topographic maps, and just doing ground searches to try to find potential habitat. But after those rounds, I wanted to try to improve our searching and kind of zero in on possible habitat zones. So I put together a habitat model with a few parameters. And it's a work in progress. This is the 3rd version of it now. But I used our known sites as a basis for the model parameters. And so nearly all of those had a slope of less than 11 degrees. I used NDVI, which is a normalized difference vegetation index, which uses Landsat data. It's a reflected visible and infrared light, and it provides a measure of vegetation type and health and general ground cover. So it ranges from like snow on the low end of the scale to, which is highly reflective, to forest at the upper end of the scale. And I used our known sites to identify this NDVI range that represents that vegetation. And then for soils, so far I just identified them as being volcanic. I looked at some other soil and geology GIS layers with things like water capacity, drainage type, and texture. But I still haven't really found a characteristic that's more specific across all the sites, but still leaves out other areas where we didn't find it. So maybe one of the geologists on this call could help with that. But I also used a land use GIS layer to select just rangeland areas, so that just excluded developed areas, forests, and lakes. And I've also looked at using precipitation elevation, but I've left those out of the model at this time. So this green layer here is our habitat model, and the blue pins are the sites that we use to develop the parameters. And the model helped us find this cluster of sites around Highway 66, kind of in the middle on the east side of the monument, which included those new Aspergillia californica var. Gigania sites. And going forward, it looks like that southeast corner of the monument, kind of near Jenny Creek, might have some good potential habitat. And the California side of the monument also seems like it should have some decent potential habitat, but again, that wild horse disturbance is an issue, and then access is also a big issue down there too. And there's a ton of really good looking habitat outside the monument, especially to the southeast there, but most of that land is private. And so after we finished our first round of field work last fall, we wanted to genetically confirm the ID of the Dimetacarpon species that we were finding. And so Andrew and I took specimens from each of our first 4 plots back to the Calabria lab at Evergreen, where we extracted their DNA and amplified the ITS and LSU regions. And our sequencing results confirmed an ID of Dimetacarpon bachmanii. Several of our vagrant populations were discovered later in these subsequent rounds of field work, and so we still need to complete sequencing for those, including the 4 Aspergillia californica var. Gigania sites.
And so here's an example of some preliminary statistical results that show Dimetacarpon bachmanii's ground cover percentage between sites of different grass dominance. We found a significant negative relationship in Dimetacarpon abundance when non-native grass dominated a habitat, providing evidence that more non-native grass management is probably needed. We also found significantly higher grass coverage in plots with invasive grasses, showing how these grasses have much thicker growth. These habitat differences between plots may affect the suitability as a vagrant habitat.
So thanks to a research grant from the California Lichen Society, we also expanded the project into Northern California over the past month. And we located and documented several new Dimetacarpon, vagrant Dimetacarpon populations, as well as a new population of the more common Aspergillia californica. And so the overall study now covers 12 plots in both Oregon and California. And so these points on the right on this map over here, they are not all vagrant populations. These represent both positive and negative data points. So I wanted to try to refine the habitat model a bit. So kind of everywhere that we went, we took pins, both of where we found vagrant populations and likely habitat, as well as open areas with poor habitat where the model might be likely to include, so that in the future I could try to exclude those areas, look for characteristics that might exclude them. And yeah, go ahead, Andrew.
Oh, yeah, we're also interested in the wind dispersal mechanics and the water utilization kinetics of the vagrants, and are currently formulating experimental pilots investigating these 2 aspects of vagrant life history. And to try to understand the reproductive strategies and individual diversity of these vagrant populations, we believe it's important to look into the population genetics as a possible future project. And that concludes our report. We would like to first and foremost thank the Friends of the Cascade-Siskiyou National Monument for their financial support, as well as the Evergreen State College and the California Lichen Society. Personal thank yous go out to Michael Zerpoli, Joe DeMeglio, Stephen Sherritt, Family Villela, the Vesper Meadow Restoration Preserve, Brit's 1998 Toyota Corolla DLX wagon, and all our friends at the Northwest Lichenologists. And here are our references. Thank you very much.
Thank you.
Thank you, Britt and Andrew. I also liked the font, looked a little Old West on your presentation. That was fun. That'd be cool to have a movie about lichens. I will hand it over to Kaylin. I know there's a question in chat.
Thank you so much, Stassi. The question we have for you at the moment, and again, if you do have a question, please put it in the chat for me. Do you think the rarity of these species reflects an actual rarity or range restriction, or a scarcity of surveys in the habitats these species appear to favor range-wide? Based on your presentation and the habitat suitability model you made, it seems like perhaps they were undersurveyed.
Yeah, I would believe that these are pretty much undersurveyed, and it's not. I'd like to be a little clear. It's not that the species are quite rare. Maybe the Aspergillia gigantea, California var gigantea is rare, but the other ones aren't rare on a species level, so to say. But the morphology of certain thalluses are rare,
especially in the monument area, like west of the Cascades crest, because you'll find more of them farther east. But yeah, especially with the Aspergillia, they can be so hard to see that I would believe that, you know, they would have been missed because we hadn't really looked for them before. But once we went to the type locality, we kind of had an eye for them, and then we found several locations within just a couple of days. So it's very possible that it's more, that species is more widespread as well.
Yeah, I think, you know, like, I feel like lichens are probably a little overlooked anyway. And these types of lichens are kind of the oddballs of the oddballs. So not a lot of people know about them either.
Okay. Thank you so much for that. And another question for you. It looked like a lot of the data points were on the edges of open areas. Was there any investigation into the center of open areas?
Yeah, I think we covered all the open areas. We were especially concentrated in the center of these open areas. We weren't really targeting edges that much, but
yeah, a lot of those open areas, it's hard to tell from satellite, are pretty well grazed as well. And that definitely affects the occurrences of these populations.